the associate professor with tenure at Washington University School of Medicine in St. Louis. I met Jeff we were putting together a grant application through another good friend of the UFDI, Hubert C. And then it turned out that Jeff and I actually grew up about 25 miles from each other in rural eastern North Carolina. He's actually more rural and more eastern. You'll have to, his accent. So Jeff is an international expert in beta cell biology and differentiating stem cells and beta cells. He's really unlocked a number of key events that allow the successful differentiation of beta cells or differentiation of stem cells into beta cells. And in fact, I'm on calls a lot for this Human Island Research Network, which is the main part of Endocrinology. And they're always talking about Jeff and Jeff's protocol. So it's good that we have a grant together, then I can beam with pride. So with that, thanks for visiting us. I'm sorry that I'm a bad driver and got you in a car wreck yesterday. But other than that, I hope that no whiplash has caused problems with our seminar.
Well, if I stutter or anything, it's completely your fault. And Clay wasn't he was rear-ended, so it wasn't, all jokes aside. Great driver, maybe needs better situational awareness, but he was not at fault for what happened here. So I appreciate the opportunity to be here. I don't think I've actually been to the state of Florida since I was a child, though I can go to Disney World quite a bit, and I'm thinking about doing that with my own 6-year-old here very soon. But I've obviously been very aware of the amazing work going on at the Diabetesinstitute here, and especially on the work going on with the biomedical engineers associated with it as well. With me being a chemical engineer by training, turned into a stem cell biologist. So it was great to actually see the vibrant and interdisciplinary environment that you guys have here with my own eyes, and I can understand why there are so many great discoveries and advances that occur here. It's just a natural consequence of having such a great environment. So something that I'm glad to have seen, and I got to meet a good number of you and a lot of the trainees as well earlier today. So I do have some disclosures I need to get through for CME purposes, I believe. So I have some patents on what we're talking about here, and these have been licensed as biotech companies. I'm also currently employed at a biotech company, have some stock and stock options there. And my wife has employment at a company that isn't relevant here. So I'm not going to be talking about any clinical recommendations. This is all research stage stuff, but I got to cross my I's and dot my T's, as people like to say, and I know that's backwards, it was a joke. But anyways, getting to the actual meat of what we're going to be talking about here today. So these are the broad sorts of questions that I'm going to be focusing on here. And I'm going to admit that especially some of the questions on, how do we mature cells? We don't have full answers to these right now, but I'm going to be doing my best to be sharing with you kind of what's the status of the field in general, and the status of what my team has been working on for the better part of a decade in this space. So I alluded to this, but I'll talk about it a little bit more. So I'm a engineer turned stem cell biologist, and that's reflected in the composition of people in my research team at Washington University, where the team is half biomedical engineering in terms of background or current program, and half, some version of biology. And this is done very purposely. That I really believe in the idea of bringing in different ideas, different expertise in order to ensure that we're not only trying to address the most pressing and important questions in the field of regenerative medicine and type 1 diabetes, but we're also doing so in a way that is most efficient and most productive. And it is, this sort of multidisciplinary environment being in the same research lab is just a very vibrant and fun place to work. And I'm very happy to be working with these fine people every day. So these intro slides are probably really not necessary to go be very, very quick about them, which is to first remind you that diabetes is caused due to the loss of healthy beta cells that can maintain normal blood glucose levels in patients. And the loss of these beta cells can occur through various different mechanisms, with the one that I tend to think about and talk about, but the most being in type 1 diabetes, for which the immune system has destroyed these beta cells. But kind of regardless of the underlying source of the cell destruction, patients who have diabetes simply just don't have enough healthy beta cells to control blood sugar levels. Fortunately, we have this amazing life-saving therapy, one of the greatest advances in human medicine available to treat these people, which is insulin injections. Is, again, life-saving therapy, really big deal. I'm saying that just because I'm going to talk about something that's going to be a competitor to that, so I don't want people to think that I think this is a bad idea. Obviously, it is not. And with the advances in pumps and sensors and technologies of the sort, the utility of insulin therapy is only increasing by the day. But this is only a treatment, and it's also a very imperfect treatment in a lot of circumstances. That people who are on insulin therapy can suffer from a wide range of complications, and have, on average, have an increased risk of death and lowered lifespans. And the reason for this is that, and this may be, preempting a little bit of the motivation for why we're focused on stem cells here, is that it's basically impossible with sensors, pumps, and human intervention to do the job insulin-producing beta cell is normally able to do. And that's essentially what insulin therapy is a manual recapitulation of what a beta cell does in the body. And this is just really hard to do with current technologies. And that results in there being imperfections in the control of blood sugar in these patients that are a major source of the health problems and complications that patients have on insulin therapy. And so the idea that motivates my lab, and obviously a lot of labs across the country and a lot around the world, is into essentially source replacement cells and tissues exogenously to replace a function lost in the endogenous tissue. So instead of manually doing blood measurements and injections, or using sensors and pumps to do that, let's just actually get the real thing, put it back in the body, replace and increase the total number of healthy beta cells available to control blood sugar levels in these patients. And of course, this is something that is done at a frequency, not super common, obviously, in the field, but this is done with isolated islets that are sourced from deceased donors. And this is a example from a review article that some colleagues put out a few years ago that I think really just illustrates the power of cell therapy here. And this is a type 1 diabetic patient who was on insulin with just very poorly controlled blood sugar levels. You can see on these CGM measurements. After a islet transplantation, you can see that blood glucose is pretty stable throughout the day. So this, again, powerful proof of concept that cell therapy can work. Again, this is done with deceased donor tissue, and which is something that is of limited supply. And when you look worldwide in terms of the total, this is total diabetes, not just type 1 diabetes here, but it gives you a sense of how many people worldwide have diabetes, and which is in the hundreds of millions. And if we want to begin to have cell therapy being accessible to any reasonable fraction of people who could benefit from it, that tells us that we need to have a large-scale sustainable source replacement cells and tissues in order for that to happen. And so with that motivation is really where I started in late 2000s, early 2010, 2011 period, working on using stem cells as a potential renewable source of cells for cell replacement therapy here. And so we're using a type of stem cell called a pluripotent stem cell, which is different than MSCs, different than hematopoietic stem cells, for example. These cells are able to become any cell type found in the body. And so that includes producing beta cell. This includes heart muscle as well. And like I said, literally every cell found in the body. And the way this works is that if you give these cells the correct cues in terms of what factors you add to the cell culture media, these cells can undergo a process known as self-renewal. That is, they can grow, they can proliferate, they can divide, and stay as pluripotent stem cells. And these cells have a virtually unlimited ability to proliferate and maintain their pluripotency. If, however, you give them a different set of cues, they could be essentially kicked out of the self-renewal program into a differentiation program. And again, depending on the specific compounds you put into the cell culture media or other factors you may use to induce differentiation, you can then influence them to become beta cells or other cell types, depending on what your cell type of interest is here. So I'm gonna make sure I remember what this looks like. So we have a few protocols published at this point that are capable of making these cells either completely in like spinner flasks or bioreactors, or our more preferred approach, just because it's easier to do this in an academic laboratory, is to do the differentiation process in high surface area flask, and then in a reactor or just a shaker plate, aggregate the cells. But the point here being that there's a lot of variations on how to make these cells. No matter which variation you're talking about, the final product here are three-dimensional clusters of cells that are roughly the same size as isolated islets. So these are about 150 micrometers in diameter. Many of the cells which are capable of producing and secreting insulin. I just have a red fluorescent protein there showing they're driven by the insulin gene to show that fact. And so I want to spend a little bit, when we talk about here, a little bit about the history of developing stem cell-derived islets, what are the challenges along the ways, how do we overcome it, before I get into some of the latest data here. And we talked about this a little bit at the training lunch, actually, so this should be a good reinforcement of that discussion. In the early days of trying to make stem cell-derived islets, the field was very quick in being able to get essentially halfway there. That from developed biology studies, people had figured out the signals necessary to take these stem cells, which are kind of similar to inner cell mast cells, so like a 200-stage embryo stage where they're still pluripotent, and we're able to get to pancreatic, what we call pancreatic progenitor cells. These are epithelial cells that are highly proliferative still, that express PDX1. So that was figured out pretty quickly because we had a good understanding of how that works during normal embryonic development. But if you kept on trying to push these into beta cells, instead of producing a beta cell, you would instead produce endocrine cells, but these were polyhormonal cells. So they would oftentimes have insulin with glucagon and sometimes also somatostatin in the same cell, and they were dysfunctional. They couldn't secrete insulin in response to a glucose stimulation, and in fact, they secreted orders of magnitude lower insulin on a per cell basis than what a real beta cell could do, and also just weren't useful for therapy in general. But we did know that these kind of intermediate cells were indeed had the potential to become a beta cell, because we transplanted these into the black box, which is the mouse here, and waited about half the year. Some fraction of these cells could spontaneously become cells that resembled a beta cell. So of course, from a therapeutic perspective, this approach has a lot of downsides to it, and clinical trials have shown it's been very difficult to get to work robustly in a human context. Mice work well, but humans do not. So here's just some internal data from my lab just showing it working pretty okay in mice. Again, about half a year later, we can get pretty decent human C-peptide levels coming from the spontaneous in vitro differentiation process in mice. And so when I started working on this full time, starting about 2011, the question that I was facing was what are the signals necessary to correctly specify beta cell identity and also get these cells to function in any degree of maturation in vitro. And so I'm just going to give you the answer. Here's the answer. And so basically, and again, we talked about it a lot during the trainee meeting, and it's not exactly the most glamorous workflow that we ever did here, that we basically scoured PubMed, found pretty much any paper that was relevant to trying to make these cells, bulk compounds based on them tried them out empirically, did a lot of combinations of the cells empirically, and just in a very iterative fashion, figured out what the correct timing of the stages were, what the correct markers should be looking at, and one of the important ones is this combination of PDX1 and NKX6-1 being in the proliferating pancreatic epithelial cells before we induce into a endocrine cell here, and really understanding that it was not just single factors that were sufficient to make any of these cell types. It was really the combination of multiple factors in order for us to have a process that had any degree of robustness that we thought was sufficient to report in the literature. And really the combination of these three factors here, thyroid hormone, a gamma secretase inhibitor, and a TGF-beta receptor 1 inhibitor was the key combination that was necessary for us to succeed. The other factors here, typically were able to just increase the efficiency by 5% here or there. So a very iterative, non-glamorous approach, but just because of the deficiencies in what was known in the developmental biology literature at the time, it was really the only approach that at least I could think of in terms of how we should make this happen. And there's just some data from the publication. It's the only data set I have for this early work, just showing that these stem cell-derived beta cells can co-stain for C-peptide, which is of course produced by the insulin gene, and other markers of beta cell identity, such as NKX6-1. Similar to the primary cells, and these early polyhormonal cells were not able to do that. Again, they could have C-peptide, but they would lack other features of a beta cell, such as expression of NKX6-1. We transplanted these into an Akita model of diabetes, and we're able to have restoration of blood glucose levels here. I think the data here is a glucose tolerance test showing this data. Again, using the polyhormonal cells as a control, super high glucose levels throughout the entire study. Our stem cell-derived beta cells are able to, the mice receiving these cells are able to tolerate glucose quite well, though not quite as good as an equal number of primary human cells that we transplanted as a sort of positive control here. So this showed us that the cells indeed do work, can control glucose, but we're still having deficiencies in their functional maturation when it came to the real thing, this being islets sourced from a deceased donor. This did end up going on into clinical development and is currently subject to a clinical trial and has been dosed into a small number of patients at this point. So this is all like in progress press release stuff here. So I'm telling you non-peer-reviewed thing just for a moment, but I think it's pretty cool. This is a New York Times article on the first patient to receive these cells, who, as of the last update I've heard about this, is off of insulin with his blood glucose being completely controlled by the stem cell-derived islets produced with this process. And so after this, I ended up moving to St. Louis, working at Washington University, and I had several immediate goals in mind that I wanted to tackle. And there were certain deficiencies in the process of making these cells that I wanted to overcome. I already mentioned, one, which is the cells were not as good at cell therapy versus primary islets. We also had some issues with there being batch failures in the manufacturing. About a quarter of the batches would just fail spontaneously about halfway through the differentiation process, which is quite annoying considering the high cost of the growth factors necessary to grow the stem cells and do the differentiation. So I just wanted to improve the cell product that we're making here. I also wanted to see if there was any new biological insights we can get from this, because now we had essentially a model, an in vitro model of human development, and we wanted to see if there was any new biology that could be gained from studying such a system, which could then itself be used to feed back into improvements in the differentiation process. And so we spent a lot of time, and this was led by a grad student who graduated from my lab and a post-bac who's gone on to grad school and since graduated, hence the double doctors here. But they went and started looking at optimizations of the differentiation process, particularly looking at stage 6 of the process, which we, the way we think about this is that stage 5 specifies beta cell identity, but stage 6 is there really to give the beta cells the signals and nutrients and stuff necessary for them to mature and be able to secrete insulin in response to glucose. And so several changes were made. I summarized it here, but I think there was 26 changes in total that we tested on a individual and combinatorial basis as well. Again, mostly in building this serum-free media to make it, which we were using a standard islet serum containing media, which turned out to be quite detrimental to stem cell-derived islets. So we had basically came up with a better maturation media for the cells to sit in for a couple of weeks during stage 6 in order to improve their features here, which I'll show in just a second. But again, like this new process now, again, still completely in 3D suspension, still made these islet light aggregates for which, we have plenty of beta cell markers and still have some of the other hormones being expressed there, such as glucagon and somatostatin, which I don't have a picture of here for some reason. We have, a whole panel of factor or markers that we look at to characterize differentiation batches here. Again, these are kind of usual suspects for islet identity, just showing that these stem cell-derived islets are, containing many of the markers you would expect to see here, either in the beta cells themselves or again, the glucagon and somatostatin expressing cells that are present at a lower fraction of the beta cells we are making here. But the main thing we really cared about was the function of these cells. Again, in vivo and in vitro function of the cells were not as good as primary human islets from deceased donors. And that's still true today, but we've done a lot of work to really close the gap in terms of their in vitro and in vivo function. So this is in vitro functional data looking at from the original protocol in 2014 versus this is a publication from 2019, which kind of shows where we're at currently in terms of function, and just showing that indeed the later versions of the protocol are able to not only have a higher amount of insulin per cell, but also tend to be more glucose responsive versus the older protocols, and are starting to approach batches of primary islets here, but are definitely not as good, still not as good on average, and not as good as some of the higher performing islet isolations that we're able to get our hands on here. And they're also able to respond in a kind of a stepwise fashion to glucose levels. But one of the greatest, one of the biggest features that the protocols were able to of beta cell identity that the protocols were able to start to recapitulate here was dynamic function. So the original 2014 protocol was unable to mount really any meaningful first and second phase insulin secretion response to high glucose stimulation in a perfusion system. Again, now with 2019 and subsequent protocols that are a little bit better, we're able to get definitive first phase and second phase responses here, but again, not quite as good as cadaveric human islets. And that's a line you're going to be hearing me say a whole lot during this presentation because it's a very important nuance that is sometimes glossed over here. So again, the cells weren't perfect when we first made them, but we went from nothing to something. Publications from my own group and other groups, again, have studies that they've done as well in their own version of the protocol, are starting to continue to, or continuously closing the gap here, including, getting, features that you expect a real beta cell to do, such as in this case, dynamic function. And one thing I realized I don't actually have any slides here because I could see the next one on the screen down there. But one of the things we did in this follow-up study was to figure out that this acquisition of dynamic function was completely controlled by this transcription factor called 6-2. And we found out if we knocked out that transcription factor that we could still indeed make beta cells, but they had zero ability to respond to glucose. And our protocol, our newer protocols seem to be inadvertently, because we didn't know about it when we were making the protocol, was inadvertently turning on that gene while it was actually repressed in the earlier versions of the protocol. So kind of the summary here is that indeed it's both a temporal and a combinatorial way of thinking about things that's necessary to make these cells. You have to know what are the factors, know the combination, and know the correct ordering here. You start switching things arbitrarily, we hit the timing off by like a day or two, then the entire differentiation process can fall apart. And it's really that sort of precision and sophistication that's necessary in order for differentiation protocols to work to any rigorous degree. These cells are glucose responsive, but again, still not quite as mature as real beta cells. So we next started thinking more broadly about the cues that the cells are responding to in order to become stem cell-derived islets. And basically all the work I've shown you so far has been focused on kind of a more traditional stem cell biology view on things. You add in small molecules and growth factors that target signal transduction pathways in order to instruct them to become beta cells. And that's about how dev bio textbooks teach things as well, is how stem cell biology protocols are usually developed. But these are all in the broad category of soluble factors. And I wanted to start using our biomedical engineering toolbox a little bit. And so we started asking questions about how the insoluble microenvironment could be affecting this. And this is known to be important in some other systems. I think about the classical system is MSC differentiation is largely influenced by the stiffness of the substrate they're on, and like soft substrates can make fat cells, stiff ones can make more like bone and muscle. But that was largely absent as a concept in pancreatic differentiation. We did know a little bit though. We did know that the way that if I can find my pointer, there it is, that the way that the three-dimensional aggregates are put together can have a big effect on the function. And we were already dispersing and reforming aggregates at the end to make sure they weren't too big in order to maximize their function. But largely, it was unknown how the insoluble environment influenced this. We also had another hint, which is, again, all the protocols I talked about at this point were all done in 3D systems. These are suspension cultures with the cells grown in aggregates. We did know that if you had the cells like in a petri dish, just played it down in the monolayer, you did the protocol, you can get about halfway through the protocol, and then it wouldn't work anymore. That there was some sort of overriding signal from the petri dish that would override the beta cell induction cues we're giving them, such that you couldn't produce any meaningful number of beta cells if you had them attached to a plate. So one of the first things that we asked here was, is it possible that the stiffness of the plate itself could be a cue that is inhibitory for making pancreatic endocrine cells? And so we did a fairly simple experiment here where we varied gelatin, collagen gel height, where there would be a correlation with greater thickness of collagen gel, which is quite squishy, with a decreasing stiffness that the cells would experience. And indeed, we're able to see that there was a correlation between the gel height and the expression of endocrine genes, as shown here. So it did seem that at least stiffness seemed to be having some sort of role in what was going on in the differentiation process. And from what we know from a great wealth of literature out there when it comes to stiffness and cellular behavior, one of the main ways that a cell is able to sense that they're on a stiff substrate is through the actin cytoskeleton, where if the cells are sitting on a stiff substrate, the actin cytoskeleton will be very well organized, very polymerized, which you can see here, for example, on the left of the screen for a slide of my postdoc put together from his grad student work days. If the cells are sitting on a soft substrate, you see a lesser degree of this. You get less F-actin as the polymerized form of actin, filament actin, and it's not well organized. And depending on how well organized and polymerized the actin cytoskeleton is, this can influence the phenotype through a large number of mechanisms and signaling pathways, such as YAP signaling. And so kind of thinking about this and really wanted to have a process that could work more robustly and kind of work at scale as well, we came up with a very small curated compound screen of compounds that either directly affected the actin cytoskeleton or affected related processes or signaling factors associated with that. And we got a hit, which is the trunculin A, to be a very strong inducer of endocrine gene expression. So the trunculin A is a compound that depolymerizes the cytoskeleton. This is just showing it with some of these pancreatic cells. So the trunculin A, the cells maintain PDX1, but you lose the polymerized form of actin This is quantified over here with Western blots. And indeed, if you give cells this compound, you were able to induce gene expression not only by PCR, but you can see that with protein markers such as from neurogenin 3, which is a very important transcription factor to induce endocrine cell fates here. And this was a dose-dependent effect that we saw here. So this was something that we were not really expecting. We were actually trying to go more for a tissue engineering angle here and kind of stumbled upon the observation of what we were seeing here. So we wanted to understand a little bit more about why this was happening, because we just thought it was such a odd result. I don't know if I would have ever predicted that polymerization or depolymerization of actin would have any influence on endocrine induction. And so when you have a weird biological system and you're really surprised by it, you want to understand some more, what you do is you turn to single cell RNA sequencing. So that's exactly what we did here. So we took our pink-hearted progenitor cells and either kept them untreated. These are all, again, still all plated down, kept them either untreated, treated them with a trunculin A to depolymerize the actin, or we gave them a different compound called nocladazole that causes hyperpolymerization of the actin cytoskeleton to kind of see, to get an idea of what happens if we go the opposite way in terms of cytoskeletal state. And so from this is, again, single cell RNA sequencing, dimensional reduction using TSNEs, because it was a few years ago before UMAPs were very popular as the way to visualize this information. We were able to identify, yeah, I do have that there, identify progenitor cells, endocrine cells, exocrine cells, and just showing some of the markers to make you hopefully believe the designations that we gave these cells here. But because of our ability to track what conditions these cells came from, we're able to go back and look to see what treatment groups resulted in the contribution to each of the cell identities that we were able to identify here. And so plated control, maybe unsurprisingly at this point, was able to show the majority of cells being progenitor cells still. Trunculin A thankfully showed that we're making endocrines. So it's again another confirmation of the data that we're showing with the bulk assays. But now if you treat it with nocladazole again to now hyperpolymerize the cytoskeleton, you now go the opposite way in a sense and are now making cells that have exocrine markers here. So this gives us a kind of a new understanding of the microenvironment where if you want to make progenitor cells, you need kind of a Goldilocks level of actin polymerization to achieve that. If you want to make endocrine, you had to depolymerize to some degree the actin cytoskeleton. But if you want to make exocrine, that's promoted by hyperpolymerization of the actin cytoskeleton. So it's kind of a Goldilocks sort of situation here that you got to switch from one porch to another. You go from progenitor to a endocrine cell. So this allowed for us to build our understanding of the signaling pathways involved in differentiation here, starting to really fill out the insoluble axis for what's going on here. As probably is obvious, there's a lot of space here that is still not filled with understanding, and that's a very active area of research in my lab right now. I'll preview that this is not the only place in the protocol where the insoluble microenvironment has an effect on the differentiation and can be modulated to indeed improve the differentiation. So we're really hoping that by continuing to study not only the soluble but the insoluble microenvironments, we can continue to edge forward both our biological understanding of cell fate acquisition, but also protocols that can be used based on this understanding. And now this gives us the ability to indeed make beta cells without having to have the entire process done in a bioreactor or a spinner flask, which is pretty a headache to work with. Yes.
The pH of the insoluble microenvironment at all?
Not directly. I mean, we've made some observations about the pH changing during the differentiation based on like the changing of the phenol red in the cell culture media that we use. We've also done some measurements of lactate production, and that's extremely high during the first like 20 days of the protocol, and then shuts off really quickly when you switch over to endocrine and beta cell fates there. But precisely your question, no, we've just been kind of dancing around it. But I could imagine pH probably has a effect on it, and we don't have as good of an understanding of that as I would like. Moving on to some more in vivo data here. So I referenced that now we can do differentiations in the attachment culture. So we've been doing that's mostly what we do at this point, just because we find it easier in the beta cells to be of higher quality when we do that as well. Here's just some evidence of that. This is making of SC islets using with the cells being on plates, using Latroncamin A as one of the factors for endocrine induction here. This is actually summarizing transplant data from 2 different publications, just to kind of show the reproducibility here a little bit. So these are mice that were induced to be diabetic. We made SC islets either from this ES line or from this IPS line, and monitored restoration of low glucose levels here. You can see, plus or minus like a week or 2 here. There does seem to be some temporal variability in our animal studies that we've seen that indeed we're able to drop into a normal glycemic range within a few weeks, no more than a month here, at a rate that is similar to now a comparable number or same number of human islet, cadaveric human islet cells as well. So we've been able to bridge the in vivo gap in terms of function to being not as good as primary islets to being comparable to human islets now. But of course, we're going to grow a unlimited number of these cells in a reactor, which is useful than having to wait from IDP or protolabs so that you know when islets are available. So that's it's been quite good for studies that we've been doing right now. So kind of the summary of part 2 here is that it's not just the soluble, but it's also the insoluble environment that can have a very large effect on differentiation. In particular, we found that the degree of active cytoskeleton polymerization influences progenitor versus exocrine versus endocrine cell fate, and that these cells generated through cytoskeletal modulation are able to rapidly reverse pre-existing diabetes. So going back to this data that I showed you just a moment ago, one of the things that we were very curious about was what is the nature of the in vivo temporal aspect of what we're seeing here. We know for progenitor cells that that is a spontaneous differentiation system. You go from a progenitor cell to beta cells. That's it is a major transformation of cell states from being uncommitted to being committed. They've been well studied. We weren't really certain what was going on here because these cells appear to be terminally differentiated. They weren't really proliferating. There's no more than what a primary beta cell does. So it was kind of a mystery in our mind. We did know some other things here, that it's not just like random measurements of glucose that are improved with time of the transplantation with these cells, but also glucose tolerance is improved, and C-peptide and insulin levels are also increased as a function of time. And we usually stop our studies at about the half-year mark, and you can see, plenty of endocrine cells there. In unpublished studies, we've gone for an entire year, which is the extent our IFIC protocol allows for us to do with, and there's no sign of these cells slowing down. And so we suspect it's probably like a wound healing response and vascularization, but 2 of my grad students at the time really wanted to know, is it possible that these cells are actually undergoing maturation after transplantation? And so in order to study this a little bit more robustly than the tools we had at the time, we ended up turning again to single cell RNA sequencing, and basically spent the entire summer trying to figure out how to get these cells out of the mice, purified enough and then getting high enough RNA out of them in order to run single cell RNA sequencing here. And this was a very very technical blood, sweat, and tears sort of approach, and very tedious in order to make it work out quite well. But by the end of the summer, we were able to do this. And I'll be showing you the data here in just a second. And so we did this with both an IPS line and also an embryonic stem cell line. looking at cells in vitro and then after being transplanted for 6 months here. Now, these are again the UMAPs showing the ES cells and IPS cells. We could again identify cell types here based on common markers used in the literature for a cellular identity. And so I'm going to be focusing just on the beta cells here for time, but this is something that is published. So we do do this analysis for the other major cell types as well. So we look very globally here at the beta cell population for the in vitro SE beta cells compared against the primary human beta cells versus the transplanted 6-month transplanted beta cells here. You can say in general, with this highlighting the most differentially expressed genes here, in general, the grafted SE beta cells look similar to the human islet beta cells. This is also supported by a Pearson correlation here. So we do see, it does seem globally there is a shift for the cells to be more similar to a primary human islet. We look specifically at the cells, the genes that change from transplantation. We had looking before and after transplantation, we had less than 1,000 genes we found were different. But the genes we saw that were different did make a lot of sense and pointed towards maturation. The genes that went up with transplantation are associated with function and maturation, such as IAPP, MAPA, GCPC2. The genes that went down with transplantation or immaturity genes, such as the other hormones or FEV, which is a very early progenitor marker of beta cells. So this indeed looked like that we are having maturation here, and we have that traditional literature published in the publication. This is just looking at just 3 cherry-picked genes here that and before this paper was usually asked about for our in vitro stem cell-derived islets because they were well known to be underexpressed in from the in vitro differentiation process here, and were recovered through transplantation. So the red and the blue here are the grafted cells. The little peaks there are the in vitro cells, and the green cells here are the primary cells. This is showing that for these 3 genes that are under or basically missing from the in vitro differentiation, we now are able to have an increase in their expression with transplantation, and that level is comparable to what a primary human islet is able to produce. And so the summary for this part is that indeed SC islets are able to mature after transplantation. And this is a really big deal in terms of our thinking of the underlying biology here, that this told us that these cells were not somehow stuck at a particular in a particular gene regulatory network or a transcriptional identity because of the in vitro differentiation. They still had some ability to change, in this case, change for the better, change in terms of having a more mature identity. However, we don't know back then and still don't know right now what are the signals the cells are getting from the in vivo environments to undergo this maturation status. Actually, I don't have a slide for this, but I do have a preprint out from last year where we actually did culture the cells just long-term in vitro. We cultured them for half a year and a year. And these positive features of maturation that we saw in the animals here were not recapitulated through in vitro culture. So it's not just simply a matter of time. We know there's something magic in vivo. We just don't know what the magic is right now. And so the last thing I wanted to leave you with in the last few minutes in my presentation is kind of showing a bit of a case study of us using stem cell-derived islet technology for some studies with patient-derived cells that we've been doing. And so there is an avenue of research being pursued out there, made available because of iPS cell technology that we could be pursuing as a field, autologous cell therapy for patients, as opposed to relying on allogeneic cells. And that's just because you can take skin fibroblasts or PBMCs or kidney epithelial cells from urine, if you want to reprogram these into iPS cells, differentiate them into your cell type of interest, in this case, beta cells for diabetes, and put these back into patients. And conceivably, this could be a autologous, source of replacement cells. This also allows for us to have kind of a unique window in diabetes pathology that we can also use these patient-derived cells and study them in terms of the mechanism of disease initiation. Sorry, I've been talking all day, so I got to take a break here. And so we did a few papers on this at this point. The one that I thought would be more most interesting to talk about in this audience, and I think you're going to enjoy this, yeah. We were chatting about this earlier, is some work we've done with patients with Wolfram syndrome. So Wolfram syndrome is a rare monogenetic disorder that's caused, a recessive disorder that's caused by pathogenic variants in the WFS1 gene. It's associated with ER stress. It appears that the beta cells don't process insulin well and get stressed, and a lot of them die or just dysfunctional. And so at Washington University, we are fortunate to have a Wolfram syndrome clinic that's run currently by Dr. Fumihiko Urano, and who's a physician scientist at WashU, and we collaborate on many things together, but particularly on these patients with Wolfram syndrome that he sees in his clinical practice, and he does research and clinical trials associated with that as well. So we were able to take a few of the skin fibroblasts from these patients that he collected over the years, and for which we knew the variants that were associated with Wolfram syndrome here, and other information such as when they form diabetes. And we were able to make iPS cells from them and then differentiate them into stem cell-derived islets. And so this is showing differentiations. These are unedited, which is kind of previous one what we're talking about here. Unedited cells from these 3 patients. Differentiation looking at now just dynamic function of the cells. And again, they do produce insulin. There is some glucose responsives here, but they're just generally not great responses when we compare it to like a unrelated non-diabetic control, as an example here. So showing that we can make the cells, but on first blush, they seem to not be correct. So in order for us to do more of a proper study here, we took a subset of these patients, the 2 more severe out of the 3 patients, used CRISPR-Cas9 to correct the variant in one of the alleles, and that's shown here, and did differentiations and assessments of the gene corrected and the unedited cells in parallel to try to understand in a isogenic system with just that one base pair being changed, what are the phenotypes that we see here. So again, differentiation to beta cells, we're able to see that with CRISPR-Cas9 gene correction, we could have a restoration and glucose-stimulated secretion for both patient 4 and patient 13 here. This one, we just had 2 clones that we were pursuing here. I think for the rest of the talk, I'm just talking about 2 of the clones just to be a little bit more efficient. When you look just with the microscope with the unedited versus CRISPR-corrected cells, you can see that the unedited cells look kind of ugly. That's because the differentiation process didn't work that great with them. We got lower yields of beta cells. That was across the board with all the patients. We still don't know why that is. And there doesn't seem to be developmental defects for these patients, so it might be more of a protocol issue, or maybe points to there being an unanswered question in the clinical manifestation of these cells. But in any event, we're able to quantify, see that, with CRISPR-Cas9 gene correction, we can restore, making kind of normal amounts of beta cells and other cell types as well. This is showing the Western blot showing that indeed we could restore WFS1 protein with CRISPR correction that's much higher in the stage 6 cells and the stem cells. Restore dynamic glucose-stimulated secretion here. We had single-cell RNA sequencing, which I'm not showing here, that pointed to there being potential defects in or ER stress, I should say, in these cells. So we used electron microscopy, and we're able to observe that the ER was swollen and expanded in these patient lines that was corrected with CRISPR-Cas9. We had improvements in insulin processing as well with CRISPR that correlates with the single cell data that we saw. Similarly, for the mitochondria, we had single cell data that pointed to the mitochondria having some defects. With EM, we could see that the mitochondria tended to be fragmented and that they had lower respiratory capacity as assessed by a seahorse assay. One of the cooler things here is that we were able to show that that with these patient-derived cells, by using CRISPR-Cas9, we were able to take them from being unable to control blood glucose in a mouse model of diabetes to now being able to restore, and actually much faster than usual than our other cell lines typically do, able to restore glucose control in mice, showing that the combination of a IPS technology with beta cell differentiation and CRISPR editing can be used in combination in order to reverse animal model of diabetes, potentially then leading the way to these cells being having utility in autologous cell replacement therapy. So the summary for this very last part, and I'll be able to take the questions here in a minute, is that we can combine these technologies together to indeed to such a degree that we can quite robustly reverse diabetes. We found a lot of defects in the beta cells that are a result of WFS1 variants. And again, it illustrates that autologous therapy is something that can be pursued down the line. So I've been kind of summarizing the individual parts of the presentation as I've been going through it, but I'll say in general, kind of what I would like the future impact of this work to be is that, by using, this kind of virtually unlimited cell source that we have been able to develop and improve over the near decade we've been working on it, this would allow for there to be just greater accessibility of these cells. Deceased donors are just not enough. There are other challenges that I didn't have time to talk about today in terms of making this a real therapy that can be much more widespread. But at least from the cell sourcing perspective, this is mostly addressed at this point, and that by using a successful use of a cell therapy would allow for the patients to be healthier and to have a better control of their blood glucose over the long term. So thank you all for listening. I got a lot of great grant support from JDRF, NIH, and a few other foundations, and WashU support as well over the years, which have been very appreciative of. We're using this technology now to work with Clay and Hubert Say now at Kansas University Medical Center, trying to understand more environmental factors that are going on for initiation of diabetes. And we have a lot of collaborators across the country as well, looking at we partnered with to look at other aspects of this technology can be, we think, quite useful for. And again, my team at WashU, which provides who do all the work, and I just got to sit around and give presentations all across the country, including the wonderful Diabetes Institute here. So I appreciate you all having me, and I'm happy to answer your questions. Thank you.
Thank you. That was fantastic. Thank you very much. So getting back to the F-actin assembly, so the other side of that is extracellular matrix interactions with that. Is that the in vivo piece?
Yeah, so this is my abbreviated version of the presentation. So when I talk more about it, I actually start off with the extracellular matrix, because the actual real first experiment that we did, we're trying to tissue engineer the cells. So we had a hypothesis that if we put in certain combinations of ECM protein, that would help to improve the differentiation. But of course, we didn't know which ECM protein to look at. And so we initially decided to do a screen of basically every commercially available recombinant ECM that we could find. And we decided to do that in 2D. And we thought it'd be not perfect, but allow for us to shorten the list. And we were supposed to then tether it to a hydrogel and go back to 3D. That was the idea. We never did that. And so instead, we did the experiment. We did the plate downs from our 3D process to 2D, and we saw for every one of those, we had a repression of endocrine identity and an increase in pancreatic potential identity. And this is with fibronectin, vitronectin, 10 different isoforms of laminin and some other animal-derived things as various types of controls. And that was kind of our first hint here that maybe, and what we took from that is for what we were asking at the time, that ECM composition wasn't the most important question. Instead, the plating down and then being on a plate was the most important question. So we just switched the question and started asking that, and never actually ended up going back to answer the question about ECM. ECM obviously is, I'm sure it's important. It just, wasn't the most important question at the time.
Once you fix the F-actin part of it, maybe the ECM.
Yeah, and that could definitely be true, and it's something that is one of those like ideas that gets floated periodically, and I'm just waiting for the right student to decide to
And then
embrace
the
it.
other ones, obviously, everybody has thought of it, revascularization, innovation. Those types of neighborhoods.
Yeah, so our differentiation is pretty much pure endocrine of one form or another. So we're not recapitulating vascularization or endothelial cells aren't part of this. We're not making nerve cells or anything like that. And those are natural or like, supporting mesenchymal type of cells that are there. And so we have some work going on right now trying to either do what we call orthogonal differentiation, because we can actually do the differentiation with the factors here and then genetically induce endothelial cells in the same culture. So we're trying to do things like that right now, and also some simple mixing experiments, which we found to be harder to make work well because these cell types don't like to mix very well. So we're starting to do, well, we've been doing it for a while. We have kind of a new iteration on the project right now that JDRF recently funded. But I do think that adding in missing components like that, missing ECM, missing cellular components could improve the potency of the cells. Ed?
Yeah, I mean, I do love this story. I continue on the thread. Have you actually done lineage tracing or tracked individual cells differentiating? Because could it be changes in proliferation rates as cells are committing to a differentiated state that changes their proportions?
The answer is no, we haven't done that. Lineage tracing, I've done any lineage tracing. My hesitation has been always sort of it's hard to do that in human systems, like human cell systems like this. Could proliferation rates affect it? Oh, definitely. I definitely think that that's true. Kind of more generally, I think that there is heterogeneity in the earlier progenitors. I don't know what that is, but I suspect my gut tells me there's heterogeneity in the early progenitor cells that give them biases towards cells that we want there or cells that we don't want there, and we don't really know what that is. And so kind of with Hearn right now, we're looking at this from more of a RNA and epigenetic perspective, but some of the proliferation genes can come out of that analysis. I'm open to what that is in general, but I do think that I do believe in the idea.
So my question, I'm sorry, I lost my voice, is
Try
regarding,
drinking water is what I do.
yeah, is regarding intrinsic control of beta cell differentiation. So I'm wondering, beyond the rare variant that you talked about, if you or other people have looked at more common variants, like those associated with type 2 diabetes, since there are a lot of type 2 diabetes related SNPs that are found in MODY related genes that might impact the ability of a certain person's iPSCs to differentiate into a beta cell.
Yeah, so there's been a lot of literature about that. It's not something I focus on, though. However, but people have gone and knocked out MODY gene or GWAS genes or introduced the actual variant itself. And the short answer is yes. If you target these genes, it will influence the differentiation, if not the actual functional outcome of the cells that you're producing. I'm not. There's been a lot of papers out there. I don't quite have encyclopedic knowledge about that. But the answer is definitely yes. And maybe more broadly, I'm certain that that there is signaling going on in the dish that is outside of the actual factors that we're putting onto the cells, that are also helping the cells to instruct themselves to go the correct direction. That while we're doing as close as we can to a very controlled system, there is some, either through signaling or gene expression, which can maybe vary by a variant, or maybe mutations also occur during cell cultures that can cause some heterogeneity on what factors the cells can respond to, or they're more or less prone to becoming a beta cell. That's all part of it. And I think the field in general is just getting a little bit savvy to that when it comes to the actual process. But it's definitely been studied much more in a like asking questions about the effect, like more pathological questions, and just now people were asking questions about it from like a process perspective.
Thank you.
I thought it was really interesting in your single cell data, the differentially expressed genes between those that were grafted versus not, differences in autoantigen expression. I was wondering if you, and I'm sorry if I missed this in the literature, but I was wondering if you guys did any sort of testing of, you mentioned ER stress, so inflammatory cytokines or ER stress. Do your differentiated cells produce also less like neoepitopes or like hips or drips or something like that?
Yes, we haven't looked at the neoepitopes. Some people have looked at how immunogenic these cells are. We haven't done that, and have seen that the cells tend to die less, like with the allogeneic UMC mixtures in there. And so I suspect just because they have like low MHC, based on like a B2 expression and stuff like that. That's why I suspect it to be true. But again, as the cells mature, it seems immunogenicity is maybe tied to that. And so we still we want to think about the immune system and the use of these cells for cell therapy. These are going to be tied together, and we're going to have to deal with that together. We can't just rely on the fact that cells are maybe like not as immunogenic as like what a primary islet actually is. But I wouldn't be surprised if they also process neoantigens differently. It's not what we focus on, and so I've never looked at it.
Have you looked at connectivity of the beta cells to see for increased function rather than just transcription factors or gene expression?
I mean, somewhat. I mean, we've done, hasn't been a major focus. I'm trying to remember what we actually have done. We looked at calcium imaging of the cells. We can see propagation of that. We have, I don't think we've looked, I think it's some unpublished stuff. We looked at some of like the like connexins and things. And it kind of looked okay. We didn't really think too much of it. We get like cells like David-Piston. He's looked at some of this stuff as well. So we looked at kind of casually. It hasn't been a major focus though. But I think that with some of the reforming of the clusters, that could be that process itself could be helping to allow the cells to make the proper connections, or maybe the environment itself, the in vivo environment itself, promotes that to being more like a primary islet. But and these are not like individual cells operating in isolation. It is a tissue, and that is again not something that we're as savvy about as I wish we were. Okay. Well, thank you.